Hello and welcome to another lecture. This lecture is about the second part of our cell division discussion, and this is centering on meiosis. Last time we talked about mitosis. I said at the end of the lecture that the eukaryotic cell cycle is divided into four phases. Gap 1, the S phase, which is the synthesis phase where DNA is synthesized. Gap 2 and the M phase, which includes mitosis and cytokinesis. We then said that cell division in eukaryotes occurs by mitosis in these five following steps: prophase, prometaphase, metaphase, anaphase, and telophase, followed by cytokinesis. And then I had said that one major hallmark, the most major important aspect to remember about what mitosis does, is that during mitosis, the sister chromatids are separated. And you'll see today that that is different from what happens in meiosis. We defined some terms, and we said that chromosomes are made up of two chromatids only after they go through S phase, and that the two same numbered chromosomes that come from each parent are called homologous chromosomes. So today we want to talk about meiosis. So let's look at what happens in the very first phase of meiosis, the prophase of meiosis 1. So just like in prophase of mitosis, we have a condensing of chromosomes. We have the beginning of the dissolution of the nuclear envelope, and we have the formation of the centrosome with the microtubules that produce the fibers, the spindle fibers. But what's different here is that we have now homologous chromosomes pairing up with each other. So they are moving together and they undergo something that's called synapsis. Synapsis is that they're pairing, gene for gene pairing it's called here. So that means that they are aligning along the genes on their chromosomes next to each other so that one gene, the same kind of gene on one of the homologs, sits right next to the same gene, even though it can be slightly different, of the other homologous chromosomes. And when that happens, they can start to exchange chromosome arms. So they form these chiasmata. Chiasma just means an X, essentially, like a cross. That's what is also referred to as crossover. And at that point, they can exchange DNA material. And at the very end of the prophase 1, we have the dissolution of the nuclear envelope. So an important part of prophase 1 is this crossing over. And so let's define a few terms here, just like we did for mitosis. So we have the two homologous chromosomes, and that's no different than it was in mitosis. Right? We have one that comes from, let's say, the paternal parent, and then the other one that comes from the maternal parent. After they have gone through S phase, they have duplicated chromatids, so they're called sister chromatids. So these two are the sister chromatids. These are also sister chromatids. So from within one chromosome, they're called sister chromatids. And then we have the non-sister chromatids. So these two here are, they carry the same information in terms of which genes they code for, but the genes can be slightly different. So the way that they are expressed, or exactly the way that a particular gene manifests itself in the phenotype might be slightly different. So for an example, if there was a gene that was simply only coding for eye color, we might have blue eyes versus green eyes or brown eyes. And so that would be, eye color would be the gene, but then the version of the gene would be exactly which color, whether it was blue or green. So those can be on non-sister chromatids. So then when we have the pairing of the chromosomes, of the homologous chromosomes, and this can only happen with homologous chromosomes. It cannot be happening between a chromosome 1 of the father, let's say, and chromosome 2 of the mother. It has to be the same chromosomes number 1 or chromosome number 2. So they pair, and they're forming these bivalents or bivalents, depending on who you ask. That's how it's pronounced. And they form these chiasmata, one chiasma, two chiasmata, And so that is chi is the Greek letter for cross, for X. So we have the crossing over occurring here. And then we have recombinant chromatids. And you see now they have the dark blue and the light blue, right? So this is one chromatid here, and it's now recombinant. It has recombined with the other homolog. And so therefore we have now breakpoints that that show that they're exactly in the same area in the two homologs. But now the paternal homolog carries information from the maternal homolog and vice versa. So that's important terminology to know. I also made this table here that points out a few things that are different between meiosis and mitosis. So in meiosis, as in mitosis, in prophase, we have the chromosomes condense and the spindle fiber forming and the nuclear envelope to break down. But then in prophase of meiosis 1, we have the homologous chromosomes pair in synapsis, and they do not pair in mitosis. We have crossing over in prophase 1 of meiosis, but that does not happen in mitosis. All right, so let's go through the individual phases. And you'll see that there's a lot of it that is similar to what's going on in mitosis, but then there are some important differences. All right, so prophase 1 we just discussed. Prometaphase 1, the spindles attached to the kinetochores, right? Those are the proteins that are surrounding the centromeres. And then in metaphase, the chromosomes are aligned in the metaphase plate. That is the equator of the cell, this equatorial plate here. However, here what you have in metaphase 1 in meiosis is that the homologous pairs line up as a pair. So they line up as a pair. They can either be lining up so that the paternal chromosome points to the left, or the paternal chromosome can point to the right. So you can see here that they are oriented randomly with respect to each other. That means the dark blue is on the left, and here the light blue for, let's say, chromosome number 2 is on the left, but it could also be that the dark blue for chromosome number 2 is on the left and the light blue on the right. And the same is true here, right? So it could be that these align in this way, or they could align in a dark blue, light blue way. And that, of course, makes an important difference, because if you sort them later on into different daughter cells, they can either be mixing the paternal and the maternal genome, or they can keep it separate. We'll talk a little bit more about that later. And then in anaphase 1, we have the separation of the homologous chromosomes, right? And see that they still have their chromatids. So the chromatids have not been separated. This is the homologous chromosomes that are separated. And then in telophase 1, and then subsequent cytokinesis, the daughter cells are now ready to move into prophase 2. So that's the second part of meiosis. So what I write here is important to remember about meiosis 1 is that the homologous chromosomes pair, the DNA is exchanged between the homologs, that the number of chromosomes per cell is cut in half, and that the daughter cells are not identical. These are not identical to each other. Meiosis 1 separates the homologous chromosomes. I showed you earlier on in mitosis, the sister chromatids are separated. In meiosis 1, the homologous chromosomes are separated. All right, so then we're moving into prophase 2. So this is the end of the first meiotic division, but there's a second meiotic division. And so you see here, this is the second meiotic division, and that in itself is similar to mitosis, except that mitosis only has one phase, right? Only has one set of phases. There's only mitosis 1, if you will, but meiosis has meiosis 1 and meiosis 2. So I'm saying here meiosis 2 is very similar to mitosis. And you can see here that in prophase 2, nuclear envelope breaks down again, very similar as you see in mitosis. You have the formation of the spindle fiber, you have the condensation of the chromosomes back to the small condensed form. Prometaphase 2, the spindles attached to the kinetochore. Then you have the sorting towards the equatorial plane. And now you see they're no longer homologs, right? The homologous pairs have already been separated. So they are now no longer paired. That's what was a hallmark of metaphase 1. And now you have the sister chromatids being separated. So that is what's happening normally in mitosis, right? The separation of the sister chromatids. And you see that happening here. And then at the end, you have the telophase. And there we have a separation of the cell into two daughter cells. And the way that this graphic shows you what's going on here is that it shows you this in duplicate, right? This part is essentially the same in terms of the steps that it goes through as for each other. So these two are the same. And these two cells originated from meiosis 1, right? So if you think back at the end of meiosis 1, there's the end of meiosis 1, we have two cells. So now we're taking this cell and we're taking this cell. And that is now what you see here as this cell and that cell going through the second phase. And so that means that at the end of meiosis, we have 4 cells rather than 2 cells. Each of these cells has the same number of chromosomes. So we started with a chromosome 1 and a chromosome 2. And we're ending with a chromosome 1 and a chromosome 2, except that over here, we still had 2 chromatids that we could separate, right? And so that's what allows us to have 2 chromosomes here, and then in the end, 2 chromosomes in each cell, just because we now call this a complete chromosome, and we call this also a complete chromosome, except it has a duplication, it has the sister chromatids. The sister chromatids have now here been sorted into the daughter cells. So that's what's happening in meiosis 2. Now I want to go back here to meiosis 1, and I want to point out that we have 4 chromosomes here, but in the end we have 2 chromosomes. And that is why it's called, why I say here the number of chromosomes per cell is cut in half. Per cell, cell 1, cell 2, cell 1, only 1 cell. So there's 4 chromosomes here, there's 2 chromosomes here. That has been cut into half, but there's still 2 chromatids per chromosome. All right, so in the end, we end up with 2 chromosomes per cell. There's one originally homologous chromosome number 1 and chromosome number 2 in this case from the father, in this case from the mother, but in the other cells, the other possible cells here, we have all kinds of variation, right? We have here a complete chromosome from the father, here a complete chromosome from the mother. So they were sorted in different ways, and they were also rearranged by crossing over, and that's why you see the breakage points here with the light and the dark together now in a what's called recombinant chromosome. Okay, so those are the steps that are part of meiosis. Here's a figure that I wanted to show you that shows you another way how meiosis shuffles the chromosomes, and I mentioned that earlier on when we talked about metaphase. So I want to point this out again. In metaphase 1, in meiotic metaphase 1, that can lead to random variation between daughter cells, is what I'm saying here. And what I mean with that is the following. This is metaphase 1, this is metaphase 2, and then these are the daughter cells in the end after meiosis. So this is leaving out a bunch of steps. What I want to point out here is that in metaphase 1, so meiosis 1, you can have this arrangement where the paternals are both on one side and the maternals are both on one side, or in this particular cell with only 2 chromosomes, you can also have the arrangement where the paternal is on one side and the maternal on the other side. But for the second chromosome, it's reversed. Now you have the paternal on this side and the maternal on that side. Can you see that this is a different orientation than this? Even though it's the same cell, they've just aligned differently in metaphase. And that means when they are now being separated, this one and this one go to the left, and this one and that one go to the right. So that in this combination, you end up with the two red chromosomes in one cell and the two blue chromosomes, so paternal and maternal, in the other cell. While in this possibility, it's the other way around. You end up with one chromosome from the maternal and one from the paternal set in each of those daughter cells. When you go through the next step, and this is not even going through any crossing over, you can see that you have now this combination or that combination, or this combination where you have a blue and a red and a blue of those chromosomes. So there are 4 different combinations that can occur, but only one at a time, right? So this is what happens to this cell. Nothing else can happen to the cell after it has started to divide. I'm not saying that this and that can happen to the same cell. I'm just saying that this cell has a 50-50 chance whether it's going into possibility 1 or into possibility 2. But of course, since germ cells are produced in large numbers, certainly for sperms, you get, overall, when you look at all those hundreds, maybe thousands of, maybe millions of cells, you get the different combinations in different cells. So they can either go this way or they can go that way. So this shows you one way of how you can get random variation between different daughter cells without crossing over. Crossing over adds to the variation. And of course, if you have a cell that has not only 2 chromosomes, but let's say 3 chromosomes, then you have even more possibilities, right? You could have blue, 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 red, red, red. If this were 3. Or you could have blue, 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 and then red, red, red. Or you could have the variation on the 3rd chromosome. And the more chromosomes you have, the more different types of combinations there are with which they can align like this or like that. So with 23 chromosomes, like in the human genome, there's already so much variation possible without even having to go through crossing over. And then, of course, that happens as well. And that helps explain why there is so much variation between the way people look. All right, so when you look at the formation of the sex cells, I just alluded to that already, that lots and lots of cells are made, especially for sperm cells. Each of the cells that undergo the duplication in meiosis, for the male part, produces 1 sperm cell. But in females, that's not always the case. So there you usually have 1 cell that goes through meiosis, and then it produces only 1 egg cell in the end, 1 oocyte. While the other 3 cells that could also produce an oocyte, 1 of the 4 makes it, the other 3 usually don't make it. They become what's called polar bodies, and then they degrade. So in females, only often 1 of the meiotic cells survives and becomes the egg cell. In males, usually all survive and produce sperm cells. All right, so much about meiosis. Let's spend a little bit of time thinking about how meiosis is regulated or how mitosis is regulated. So cell cycle specific regulators allow the cell to determine whether it's even going through the various steps of the cell cycle. So we've talked about the cell cycles being the G phase, and then the S phase, and the other G phase, G1 and G2, the gap phases, and then moving into mitosis. And whether or not the cell is moving through these steps in the cell cycle, that is determined by a set of proteins that are called cyclins and cyclin-dependent kinases. So here are cyclins, and then cyclin-dependent kinases. And so they're shown here. You have these 2 different types of proteins, and they can form complexes. So it says here, cyclins bind to and activate cyclin-dependent kinases to control progression through the cell cycle. So when they bind together, they can then target other proteins, and they can transfer phosphogroups or phosphate groups. That is called phosphorylation of a protein. When a phosphate group, sometimes from ATP, when that phosphate comes off, is used, it can be added to another protein, and that oftentimes activates that particular protein. So the cyclin CDK complexes, they phosphorylate target proteins that promote cell division. So the green protein here is the one that actually determines whether or not the cell cycle will cycle or whether it will stop. And that is determined, whether or not this green protein is doing that is determined whether or not it's active, and that is dependent by whether or not it's phosphorylated, and the phosphorylation occurs through these proteins that add the phosphate group to the target protein, and those are the cyclin and cyclin-dependent kinases. A kinase is an enzyme that transfers a phosphate group to another protein. So kinases is a large class. This is a particular kind of kinase that transfers a phosphate group to another protein. In this case, it's a protein that then promotes cell division or works in the cell division cycle. So cyclins and cyclin-dependent kinases regulate the cell cycle. Now, within the cell cycle, where do they regulate the steps? It turns out that there are certain areas, certain times when specific cyclins and cyclin-dependent kinases are transcribed and become active and work in the correct timing of the movement through the cell cycle. So these green areas here are the 3 different times when different combinations of cyclins and cyclin-dependent kinases come together and regulate the movement of the cell through the cell cycle. There's the G1S cyclin, so that's the difference here between the movement from moving from the G1 phase into the S phase. The G1S cyclin-dependent kinase complex prepares for DNA replication, which of course happens in S phase. Then the S cyclin CDK helps initiate DNA synthesis. So now it's ready to move into synthesis, and now the synthesis happens. And then we have the M cyclin CDK, which helps to prepare the cell for actually now moving out of G2 and going into mitosis. So each of these transitions is characterized by what's called a checkpoint. So I say here the cell cycle is regulated by specific protein complexes made by cyclins and cyclin-dependent kinases, which allow the cell cycle to pass through 3 checkpoints. And so that's what these question marks here are. So there's certain checkpoints where something is checking whether or not the cell is ready to move into the next phase. And so at this first checkpoint, that's also referred to as the DNA damage checkpoint. This will not be passed, so the cell won't move past this checkpoint if the DNA is damaged. And so I'll show you in a second how that is regulated on the molecular level, like what proteins do that and how they can recognize that. But the cell will not go into S phase when damage is recognized by the cell. So when the DNA was damaged, for example, by UV radiation or by any kind of chemicals that destroy the DNA structure. So the cell can determine that, it has certain proteins that can detect that, and they build this checkpoint through which the cell cannot pass until the DNA is damaged, until the damaged DNA is repaired. And that makes sense to do it right here, because now we are synthesizing a new strand from the old strand. And if we're just copying a bunch of mistakes from one to the next, then that's just propagating the error. So here the error could still be fixed. So then there's a second checkpoint, that's the DNA replication checkpoint. At that point, it's determined whether or not all DNA is replicated or whether there are still some unfinished chromosomes. Of course, that's important so that when the chromosomes get sorted into the 2 different daughter cells, that all of the chromosomes are completely replicated, duplicated, right? Otherwise, we end up with more DNA in one cell and less in the other. And then we have the so-called spindle assembly checkpoint. That happens right before anaphase. And of course, anaphase is the phase where the chromosomes are separated into 2 different daughter cells. That can only happen correctly if the spindle fibers attached to each of the chromosomes, and there's no one spindle fiber that's still sort of flapping in the wind, without having attached fully to the centromere via the kinetochore. So this checkpoint checks whether or not all chromosomes are attached to the spindle. And only then will the cell go through anaphase. So let's look at one example of how these checkpoints work. And that is using a specific gene that's called P53. It's a gene that's found in many organisms and has a very specific function. So I say here specific genes coding for proteins such as P53, the name of this protein, control the activity of cyclins and CDKs. So what do I mean? Here's our nucleus, here's our DNA, and here's this protein called P53. And it's located in the nucleus. That's where it works. So when DNA damage activates protein kinases that phosphorylate the P53, then here's the DNA damage, then that detection adds the phosphate group. And I had previously said that proteins that transfer phosphate groups are called kinases. So there's a specific kinase that takes a phosphate group, for example, from ATP, and sticks it onto the inactive P53, now making it a phosphorylated P53 that is now active. And when it's phosphorylated, then P53 can act as a transcription factor. Remember transcription factors from a previous lecture? Those are proteins that bind to the DNA and they allow the recruitment of the RNA polymerase to specific genes, and then the transcription of the genes that are regulated through that particular promoter. So P53, when it's phosphorylated, acts as a specific transcription factor. It binds to a certain promoter. It allows the activation of that particular gene. And now these genes are turned on because P53 allows them to do so. And these genes that are now turned on, those are the ones, not P53 itself, but those are the genes that are now producing proteins, right? So this gene becomes active, makes a protein. That protein then inhibits the cell cycle and gives the cell time to repair the damaged DNA. So that is what the checkpoint really is on a molecular level. So this particular protein that is regulated through P53, that will now bind to the cyclin CDK, will regulate those and will now no longer allow them to push the cell through the cell cycle into the S phase. So there are a number of genes like that. So I gave you P53 as an example. There are a number of genes like that that regulate cell cycle activity. Now think about a cell that's cycling and that needs to go through regular cell cycles in order to allow the formation of new cells. So for example, during growth, you can only have growth if you have cells dividing. So you need the cells to be dividing, but you don't want them to divide so fast that it's completely out of control. And when cells grow really fast out of control, that is one of the hallmarks of cancer. So cancer is cells that are no longer regulated normally to divide at a normal pace, but they have come out of control. And so there are certain genes that, when mutated, can lead to cancer because they are no longer allowing the cell to regulate its progression through the cell cycle properly. There are two types of examples. And there's this nice little graphic that I got from the internet that I think is a really nice way to illustrate that. So these two different types of genes or classes of genes are called proto-oncogenes or tumor suppressor genes. So these two types. So normally a proto-oncogene promotes cell division. So that means, it helps the cell to normally divide, because otherwise you can't grow. You can't produce new cells. Dead cells, maybe on the skin, have to be sloughed off. New cells have to be made. Maybe there's an injury, new cells have to be made. Maybe there's just regular growth of an organism. So new cells are required. The proto-oncogene regulates it and promotes the cell division. But when it's out of control, when it's doing much more than it normally does, pushing the cells through the cell cycle, then this proto-oncogene that is here likened to the gas throttle of this race car, if it's now no longer just producing this amount of thrust, but this amount of thrust, much, much faster, and it can't be stopped essentially because it's going in overdrive, then that can lead to cancer. So in this case, the race car is balanced in its speed by the gas throttle and by the brakes. So the brakes can be put on and the gas can be put on, and so you can have sort of a regulation of growth by cell cycle division, but not too much cell cycle division. So in the case of this race car here, it drives fast enough, but it doesn't drive too fast. If it drives too fast, then you get cell cycle division after cell cycle division after cell cycle division in way too fast a sequence that leads to cell proliferation, uncontrolled cell proliferation, which is essentially cancer. The other type of genes are the tumor suppressor genes. So they normally repress cell division. They make sure that the cell does not go too fast by putting the brakes on. And when these brakes no longer work, when they're either completely gone or they're not braking strongly enough, not fast enough, then you have the same problem that you have too much cell division. You have more cell division than you should have, even though now it's not because of the proto-oncogenes. They're still allowing the regular promotion of the cell, but the brakes now don't work anymore. So you need both. You need regular gas and you need regular braking in order for this to be regulated. If you put down the gas too much, then even the brake can't help. And if you have no brake, then even just keeping the gas at a certain speed will not allow for the proper speed of the car and will lead to this over expression of cells. And that will lead to mutations that then these mutations in these genes will then lead to cancer, to overproliferation of cells. So those are the proto-oncogenes and tumor suppressor genes. So let's summarize what we talked about today. I said that in meiosis, but not in mitosis, homologous chromosomes pair in meiotic prophase 1. I said that in meiosis, but not in mitosis, paired homologs can exchange chromosome arms in a process called crossing over. I said that this shuffles parental genes and creates variation between offspring and parents. Meiosis 2 is very similar to mitosis. So meiosis 2 is similar to mitosis. Meiosis 1 is quite different. Then, just like for mitosis, I bolded a statement here for meiosis, and that is the main feature of meiosis, and that is that during meiosis, the homologous chromosomes are separated, while during mitosis, the sister chromatids are separated. So definitely something to remember. During meiosis 1, chromosomes can align in the metaphase plate in two different ways. And I say here left or right of the equator. And that was that picture that I showed you where we had the blue and the red, and they could either be blue on both sides or red on the other side, or they could be red and blue and red, so that you can either have only blue chromosomes or only red chromosomes, or as the other option, a mixture of blue and red chromosomes. So that is what I say here can happen. That also leads to different possibilities of combinations, and that the shuffling of parental genes creates variations between offspring and parents. So see that this sentence is the same as this sentence. In this case, that shuffling occurs because of the different orientation in metaphase. Here it's due to the crossing over process. So two different processes can lead to variation. And then finally, I said that the cell cycle is regulated by cyclins and CDKs that create these checkpoints, which themselves are regulated by checkpoint controlling genes and the proteins that they encode, of course. And then we talked about two different types of these genes that are regulating the normal speed of the cell division, and those are the proto-oncogenes and the tumor suppressor genes. Either one of those mutations, lack of function of either one of these types of proteins can lead to cancer. All right, and that's for today, and I'll see you in class.